Thanks, guys. I hope you can hear me online. Rob's Rob's feeling a bit tired this morning, I think, after his, this is the final day of the conference. So he's asked me to take over this morning. With that, we have a number of talks, beginning with Jacqueline Harris, identifying genetic modifiers of sickle cell disease by backcrossing genetically diverse BXD inbred strains with preclinical Towns sickle cell mice.
Good morning, everyone. I am Jacqueline Harris. I'm an instructor in the Genetics, Genomics, and Informatics department here at UT-HSC. Our chair is Rob Williams. And so the BXD part of my presentation should be explained just by me being able to mention his name today. But this project really is a cross between the sickle cell experts here at UT-HSC and the expertise of the BXDs in our department. So I am actually a novice here seeking your expert advice. So with that, we'll get started. So if I could start with a backgrounding significance of sickle cell disease. It is a group of disorders that has a mutation in the beta globin gene that promotes polymerization of hemoglobin and the sickling of erythrocytes. So the most clinically severe form of sickle cell disease is sickle cell anemia. And this is caused by the heterozygosity of the SS mutation. So clinical expression of sickle cell disease is variable. And this is because it's influenced by both genetic and non-genetic factors. So for example, high fetal hemoglobin is associated with less severe sickle cell disease phenotypes. And fetal hemoglobin is under the genetic control of different genetic modifiers. Also, if you have a co-inheritance of alpha thalassemia, then that is also protective against sickle cell disease complications like hemolysis, stroke, and kidney disease. So, sorry. The types of sickle cell disease and inheritance, again, the most common and severe type is SS. But you can also have abnormal hemoglobin C, hemoglobin D, and E. You also can have individuals that have sickle cell disease because they have a co-inheritance of beta thalassemia. So usually for normal red blood cells, they are flexible disc and they go smoothly through the blood vessel. But for sickle cell anemia, blood cells are stiff and they sickle. And this causes them to get stuck in small blood vessels and can cause pain or what is called pain crises. So this can affect any organ system in the body. So as I said, sickle cell severity is very variable, and there are genetic and environmental components that contribute to that. So again, for patients that have high fetal hemoglobin, they have less severity. There have been SNPs are associated with fetal hemoglobin, but of course, like many other complex disease and traits, if you take those SNPs and aggregate them in a polygenic risk score, you still are not explaining the variability of disease. So there is this problem of missing heritability. So there are some mouse models that are currently used to study sickle cell disease. One of them I have listed here is the SAD mouse model, and it is generated on a C57 Black 6 background. It has transgenic beta globin and alpha globin that is similar to SS in humans. And you get sickle cell features like kidney, lung, and heart function abnormalities. You have the Berkeley mouse, which does not express the mouse globin genes, but instead have the human AS and fetal hemoglobin globin genes expressed. And so these mice have a low expression of fetal hemoglobin and have a more severe phenotype than the Townes mouse. So the Townes mouse has a higher level of fetal hemoglobin, and this fetal hemoglobin goes down to undetectable at about 4 weeks of age. And then the globin A and S start to take over. So this mimics sickle cell disease in humans, which is gradual. So this is basically the interest in the Townes mouse because it's probably more parallel with human disease. So we do have a collaborator on this project at the Medical College of Wisconsin who has characterized the Townes mouse for sickle cell. And so in order to get at the genetic modifiers, we are in a good position here at the university to use the BXD system of mice. And so what this will do is allow us to build causal models of sickle cell disease and identify genetic modifiers. We can evaluate genetic effects because we'll have the statistical power to do that. So not only the genetic effects, but also environment. There is also the possibility of cell lines for this genetically diverse family to be generated. So this gives us just a good platform to be able to explore and develop a genetic population of mice to be able to look at sickle cell disease variability. So this is where I need your help. We have set up this backcross initially with a few strains of BXD to the Townes mouse. And so here you see that we're starting with breeding trios where we will have we will cross two BXD females to our town's male, which will produce F1s that would be heterozygous, but to get to the sickle cell phenotype, we need homozygotes. So we then back cross the F1s to the town's male, the father, to get to the N2. And so we expect to get 50-50 homozygotes to heterozygotes. At that point, we will be weaning the mice to do the genotyping. So the genotyping will happen at weaning. I should say that. And then at that point, once we have the homozygotes, then we can start to look at the phenomes of the mice in each version of the BXDs that we generate. So this is kind of the timeline that we have gotten approved with the IACUC. So we will start with breeding the mice. And then once we get the mice breeded, we will do the back cross, of course, genotype at weaning. And then initially, we will do in vitro sickling assays, which are hypoxia studies, get blood, do blood counts, and collect tissue. So in summary, there are a limited number of mouse models for sickle cell disease that are used to, evaluate novel therapies, but there are really few studies that look at the genetic variability of pain and organ damage in sickle cell. So this system will hopefully give us the opportunity to do that, as well as look at sex differences, age, environment, all of the above. So that is the summary, and I would like to thank Athena Davenport. She put in the protocol along with my help, so we are working together to start this breeding program, and I think the possibilities are endless on what we can do in this system. Thank you.